stage is yours.
All right. Thanks a lot, Todd. And yeah, thanks for everyone for coming today. So the work that I'm going to be showing is primarily from Dr. Walker. So all credit pretty much goes to her. And I stole a lot of her slides for this. So, all right. So how I wanted to begin is just to give a brief overview of, what Atomab is, what we do. It may surprise you, but the Upper Valley has a major biotech company. So Atomab is located in Lebanon, New Hampshire, and we do a lot of work with biotech and pharma companies all around the world. So we're special in that we don't develop our own pipeline. So meaning that we're not here to make a drug and then carry it through clinical trials. What we do is we partner with large pharma like Bristol Myers Squibb or Regeneron or Sanofi, Merck, and other biotech companies, and try to make drugs for them that then they will put through clinical trials. And so we're focused on supporting these companies in making antibodies that will then be able to go into the clinic. And so far we've worked with over 80 pharma partners, and Atomab's been around for, correct me if I'm wrong, Cody, but 13 years now. And we've done over 350 therapeutic programs. And now, to date, we have 40 antibodies that are currently in clinical trials, which is a really exciting time to be at Atomab, because we see these things start to roll out and actually really help human health. And so, to date, we have one antibody that's actually been approved and is currently in use in China. So that is it's an anti-cancer drug. And that's something that is now something you can just get prescribed from a doctor. And so over the next couple of years, hopefully more of the number of programs that are just in the clinic will be moved to fully approved drugs. And so what do we do? So Atomab itself makes therapeutic antibody drugs. And so what is a therapeutic antibody? So your body, and I'll go through a little bit how these are made, but your body makes antibodies as a way of responding to infectious pathogens. And it was realized back in the 1970s that this might actually be a way that you can target proteins to things in the body for potential therapeutic benefit. So there are drugs that are so a lot of the things that we work on are actually anti-cancer drugs. So we will make antibodies that can actually be used to bind to certain protein markers on the surface of cancer cells that specifically identifies them as a cancer, and then will target them by the body and by the body's own immune system for destruction. And so this runs in contrast to what you would typically use for like standard of care for cancer therapies like chemotherapy or radiation, which are kind of broad swords, and those are effective, but they have a lot of side effects, and they make people sick. Your hair falls out. People can often feel really, really poor with these, and they may not be as effective as you hope. The benefit of an antibody is that it sticks to its target. It's very, very specific, meaning that there are often very few side effects. And since it's harnessing the power of the body's immune system, the immune system is really good at pruning out things that shouldn't be there. And this happens in your bodies all the time, and it's why you're not sick most of the time, right? And so we're utilizing the power of the immune system to destroy things that you don't want to have in your body. And so what I'm showing here is just the basic structure of an antibody. So this is what protein would look like if you could zoom in, a million times. So an antibody is composed of two protein chains. So you have this thing called the heavy chain, which is the larger chain, and a light chain, which is the smaller chain. And that's what it would look like as a protein structure. And so these two proteins will combine to make a full-length antibody. And so there are two parts to an antibody. So this binding region, which is called a Fab domain. So on this side of the antibody is what is actually going to be sticking to the target. Here, let me put my pointer on so you can actually see. So this is what will be specific to the protein of interest. So the thing that you want to have basically targeted and killed. And it has 2 of these binding sites per antibody molecule. And then you also have this portion, which is called the FC region. And the FC is what does the business end of the antibody. So this informs the body, how do you want to kill that thing? And so the top part here, the Fab, is what's directing the immune response to a target. And then the FC portion is actually telling the body what it is that you want to do once it gets bound. And this is a really, really powerful tool for pharma in order to try to make a drug. And so as I walk through this, there might be terms that you don't understand, or I'm speaking clear as mud. So please feel free to interrupt me. I'm constantly talking to people about this, so I'm happy to answer any questions. So, especially nowadays, immunology has gotten really big in the past few months for pandemic-related reasons. So, but this is just to kind of show you the importance of these molecules in the pharma space itself. So antibody-based drugs are becoming a major class of pharmaceutical molecules. And so this field, it only started back in the 1970s. And so this differs from small molecules. Smaller molecules are, things like aspirin or like a steroid or something that could be made in a lab relatively easily, and it's not. It's more of a chemical structure and less of a biological protein. And so back in 1975, the first antibody was actually produced. So this was, the terminology is monoclonal antibody. So a monoclonal antibody is an antibody that binds to only one thing, mono for one, antibody for antibody. And so this antibody, clonal, but it doesn't matter. It binds to one thing. And so it has one target. And over time, people began to realize that this could be used as a way of treating disease. And so in 1998, or I'm sorry, in 1997, you have what begins to be some of the first approvals of drugs that become used even to this day. So Rituximab is a drug that's used for non-Hodgkin's lymphoma. This binds to a protein that is highly expressed on the surface of this particular cancer. And it uses the immune system to just basically delete all those cells in the body specifically, but you leave the other cells that don't have that protein around. And so somebody will experience far fewer side effects with this kind of a therapy. And once they begin to see the power of it, medical science really started to move this way in a quite powerful way. And you can see the trajectory over time of these approved drugs continues to increase. And this was actually, this came out in 2020. This number would be even higher to this day. And now with what has happened with the pandemic, people are beginning to really appreciate, even in just popular media, the importance of these drugs. And so, so that's kind of where things are going. But limited side effects in the body, long half-life in the body, because it's essentially a human protein. So your body will keep these things around in your blood for a really long time. And they're very potent. They bind, they stick to their thing really, really effectively. And so to date now, there are now over 60 antibodies that are currently approved and used in the clinic for a variety of different neurodegenerative diseases, cancers, viral infections, all sorts of things are now being used for antibody therapies. And so what Atomab does and what it prides itself in doing is being able to discover these antibodies. So we're able to discover a diverse panel of these antibodies that bind to whatever the target is that our partners want. And then we use extensive engineering to try to make these antibodies as stable and manufacturable as possible for people. Because it's not just, and this is something I didn't appreciate when I was in graduate school, it's not just getting an antibody that binds to the target. It's you have to be able to have it scaled in the millions of liters for eventual downstream production, right? So people need to be able to make this in large reactors that could be then used at pharmaceutical scaling. And so Atomab prides itself in being able to make antibodies that when they leave our shop, we know that this will, as long as the biology of it works, as long as it does the thing that the pharma company wants, we know that it will be able to be scaled appropriately and be able to be used as a drug downstream without any like unforeseen circumstances in manufacturing. And that's a really powerful distinguishing factor for Atomab in comparison to most other antibody companies. So now I'm going to give you a science lesson. So B-cell immunology. So these antibodies are naturally in your body, right? So everybody makes antibodies, and you've heard about the vaccines that are coming out for COVID, and we're trying to drive antibody responses against COVID for protecting you. But where do they come from? So they come from your adaptive immune system, and so the adaptive immune system in your body is something that exists in all jawed vertebrates, so from fish all the way up through us, basically. And it's composed of B cells and T cells. T cells you might have heard about. B cells aren't talked about as much, but they're really important because they're the things that are actually making the antibodies. And you have the capacity to produce an amazing number of these antibodies for recognizing what might come and attack you. So at some point in evolution, these jawed vertebrates decided that we need to figure out a way to anticipate what might attack. And so what they decided to do was to make a large number of anticipatory receptors that could actually bind to the target of interest that they might not have ever seen before, just by sheer number of options. And so the adaptive immune system has the ability to make more combinations of these receptors than there are probably stars in the sky, right? So you have the ability in your body, in this process of making these antibody genes, to produce over 10 to the 11th combinations. And so you're at a point where you're basically making as many combinations as Jeff Bezos has money. But this is a huge, huge number of antibodies that could be made by any given cell, any given B cell in your body. And this is really important because this allows you to be able to recognize whatever might come your way. So if you went into the lab and you made a molecule that never existed before in human history, and then you injected that into a person or a mouse or a cat or a dog, they would be able to raise antibodies against that thing that they had never seen before and had never existed before in nature. And that's the power of the adaptive immune system. It can anticipate what might be there and then attack it. Does that make sense to everybody?
Mm-hmm.
All right. So another important facet of the adaptive immune system is the ability to remember for the rest of the host's life. So that's why we get vaccines. The smallpox vaccine actually eradicated smallpox. How was it able to do that? Well, it was because it was a really effective vaccine that allowed for long-term memory of your body against this particular infection. And so immunological memory is in part driven by your ability to make these antibodies. And so these antibodies will provide immunity in your blood for, on the order of years to decades to sometimes your entire life, depending on how effective the vaccine was. And these are coming from B cells. So the humoral immune system, so humors, are composed of B cells and then their products, which are antibodies. And antibodies are made by B cells, and they're the thing that are going to bind to your target and kill it and keep you safe. And so that plays out for all infections or vaccinations that you experience. And so I had mentioned before that you have these antibodies that are being made, and these are being produced by a cell population called B cells. And all of this is happening in your body within defined structures called lymph nodes. So lymph nodes are essentially the reactors in your body that are producing these antibodies to then attack whatever your body is insulted by. And you can feel it when you get infected, right? That's why you get swollen lymph nodes under your neck if you have a sore throat, or under your arms if you get a vaccine. These lymph nodes are strategically placed around your body to try to filter out things that might be different, and then allow the immune system to see them and then fight them. And when you get your vaccine, that's what you're feeling. You're feeling your body's immune response against that new thing. And so how this plays out is you have these B cells. Each B cell has its own unique specificity, and we're talking, billions and billions of different specificities that can be generated by these cells. And they'll go, and in the lymph nodes, the protein or the virus or whatever it is that whatever you're experiencing that you want to have killed will be recognized by these B cells, and then they'll eat up the antigen. The antigen is the protein, immunology term, sorry, force of habit. They'll eat up the protein, and then they'll become licensed by the body to begin to produce antibody, and also become a lot higher in binding strength against the target of interest. So what ends up happening is these B cells will bind, and so you'll have these non-immune experienced cells that will eventually come in contact with the target of interest. And then those move into specialized structures that get made in the lymph node. And we don't really have to go into all of the facets of this, but the important thing is that what it does is, once a B cell is showing that it's able to bind to a target of interest, it amplifies and it begins to divide and begins to make that into a major response in the body. And as it's doing it, it actually has a way of mutating this antibody gene to try to make antibody that actually binds even better to the target. And so what this essentially becomes is Darwinian evolution, and where you have a competition where these B cells are actually beginning to mutate, divide, and try to capture more of this protein. Was there a question? No? Okay. And then at the end of the day, make really, really, really strong antibody against the vaccination or infection. And so there are two main products that come out of this, one being memory B cells. And these are cells that will go and float around in your blood for years, and they'll wait for future activation. And then plasma cells. These are the things that we're primarily concerned with when immunizing someone. This is what they study when they were studying the Sars CoV-2 vaccines. They want to have antibodies that are of high strength in the body, that can bind and neutralize the virus and keep people safe. And so these plasma cells, they actually, once they're made, they go and they live in your bone marrow for presumably years, decades, sometimes your entire life. They're just constantly there pumping out antibody, and they're antibody factories, and they're specific for that target of interest. And so this is the end goal of vaccination. This provides you with what would be called sterilizing immunity, and this is what the SARS-CoV-2 vaccination is doing. And so if you haven't gotten it yet, highly, highly, highly recommend it, because it will keep you safe. And so Atomab has basically tried to find a way to take what the body does, but do it in a test tube. And so Atomab uses yeast instead of mammalian cells. We took baker's yeast, so like Fleischmann's baker yeast, and we have genetically modified it to the point where it's able to easily make and express antibody. And then using a lot of computational biology, which I don't even fully understand, they've been able to make these large, large libraries of yeast that can express billions of different kinds of antibodies, just like the body does. And so in a tube, you'll have yeast that actually makes antibody, and it makes like billions of these unique antibodies that all would be able to recognize a unique antigen. And then what we do is we try to do something kind of like what a gold miner would do, and pan in rounds and rounds and rounds of trying to combine these yeast libraries that are very, very diverse with your protein of interest. So say this is a cancer protein, or this is a Sars protein. And then over cycles, what you'll find is that this diversity goes down from billions to maybe 5, maybe 10. And they'll bind very well to the target of interest. And then we'll test them for all sorts of different characterization for, whether or not they'll be good for manufacturing, whether they think that they would be good biologically. And it's a very powerful tool. And this entire process is on the order of months and not years. We're able to do this with very rapid turnaround, which is unheard of now, which would have been unheard of even when I was beginning my studies in college. So this has happened in very short order in the United States for making drugs. And so, Atomab prides itself on being able to move quickly. So the process itself is very rapid. We're able to get antibodies that bind to their targets that are highly specific, and we're able to find things that are very unique. And that comes from the diversity aspect of this, where you have billions of different antibodies to choose from, and we're able to pull out the ones that have the unique specificity that our partners would want. And so, that's, basically just saying the same thing I already said. So this process and workflow is something that Atomab has spent a long time developing. And so making things as efficient as possible for drug discovery was a barrier before Atomab really existed. And our CEO, Tilman Gerngross, he's an engineer. He was an engineer, and I'm sorry, he is an engineer, and he was a professor at Dartmouth, but now he no longer teaches. But he and the team here spent a lot of time in trying to figure out how to optimize this process, so that way it can be done as quickly as possible to try to discover drugs. And so this is just some data if you want to see it, but what we have the ability to do is actually make these antibodies bind better to their target over rounds and rounds and rounds of what we call optimization. And so don't worry so much about the numbers here. The point is that we have a way of mutating these antibodies, the heavy and light chains, to be able to bind better to their target. So what you're looking at here, and you can tell your friends that you have now looked at a Forte biokinetic trace, but you don't have to worry about that. What it does is it basically assesses the ability of something to bind and then also to fall off. That's it. So up here is the binding. So it binds, and then this response goes up. So you see the thing bind to the target, and okay, this is actually binding the way that I want it. And then what you do is you let it fall off, and that's measured in time. And so over rounds of optimization, what we see is that we're able to get it to bind, but then you start to see this fall off become slower, and it stays up. And at the very end, you see that this doesn't really fall off anymore. What that means biologically is that you're making an antibody that sticks to something and then doesn't come off. And that's really important for making a drug, because you want to have something that's highly specific and can be dosed at a really low level. The higher affinity something is for a target, the less of that molecule you'll have to deliver to a given patient for a given treatment, which will make their lives a lot better and also decrease the cost of treatment. And so a lot of effort in pharma is put into making very, very strong binding antibodies to their target. And so this is biochemically what we look at. You see you have your on rate and your off rate. So the ability to bind to something and the ability to fall away. And as the ability to bind and fall away improve, the affinity or strength of binding goes up. And it's a really pretty fascinating thing to watch, because at the end of the day, you make a molecule that can really help somebody, and they don't have to have like tons of it going into their system. They can have a better quality of life during treatment. So and this is sort of just a typical workflow, but we have over 250, now 350 programs at Atomab, and our goals are always to make antibodies that have a range of affinities, a range of binding properties to whatever target is delivered to us. And then we deliver antibodies back to that pharma partner. And so we usually provide them with a panel of like 48 to 96 monoclonal antibodies that were pulled out of these libraries that can then be used for their later characterization. So those get mailed to our pharma partners, and then they test them for the biological function. So they have a lot of models and in like animals like mice or monkeys or in cell lines in dishes, and they test to see whether or not the antibodies are doing the thing that they should. And then they'll come back to us and they'll want us to actually make them even better. And so Atomab then does these optimizations that I had shown you right here to try to make the drug even more potent than it had been before. And we're very, you know, not to toot our own horn, but we're very careful about this, and we've become very good at it. And so now more than 60% of the programs that we complete with our pharma partners go on to further commercial development. And yeah, I feel very lucky to work here. We do very good work. So, and this is just so this is kind of going into the other aspect of Atomab, and this is harder to show you because it's not really, these are more just like general properties of antibodies, but we do a lot of work to try to make sure that the antibodies themselves are as manufacturable as possible. And this is something called developability. So how likely would a drug candidate be developable to make it in industry level scaling? So you have biology, which is kind of up in the air, and you won't know until you test whether or not the drug that you made works. But then the question becomes, well, is it scalable? And so Atomab has a whole slew of tests that we run, and also methods that we use to try to improve the ability of these drugs to be made and then manufactured at incredibly large scale. So that then brings me to our pandemic preparedness. So Atomab has, since I've been recruited, so Dr. Laura Walker, she's kind of started this at Atomab now. I think she's in her 9th year here. So about 9 years ago, she's been kind of working on this in the background, trying to develop a way of making antiviral antibodies that may be able to anticipate when the next pandemic is. And so this was a slide that she actually presented back in 2018 to a group of fellow scientists, where she was making the point that essentially infectious diseases are now reemerging or emerging almost every year. You know, a few years ago, we had the Ebola outbreak in West Africa, and then we had the Zika outbreak that happened in South America. And at the time, she had this question mark being like, well, what's next? Well, now we know, right? SARS. So we had we had a new variant of SARS, which initially we had happened back in the early 2000s in Hong Kong and Toronto. And now we have the global pandemic of the SARS-CoV-2. And this is something that has continued to worsen for a variety of reasons. So we have globalization. So it's a lot easier for societies to interact through trade. Urbanization, people are moving into cities. The density allows for more chance of spread. Mobility, you can get on a plane in Boston and be in China in less than 36 hours. Climate change, you know, as things thaw, and as you were mentioning before, where like the sap part, like the sugaring season is actually changing by a full month since like the 1800s. You know, these are causing real changes in the way that society is interacting with nature, and it's causing more clashes. That leads to the chance of exposure that could jump species, which is likely what had happened with the SARS epidemic that we're seeing right now. And then also increased global population. The more people you have around, the more likely you are to have an infection spread. And so the point that she was trying to make back in 2018, which is actually borne out to be very, very prescient, was we're always worried about rapid response and how fast is fast enough. And so when the SARS outbreak happened, you know, that was the duration of about 3.5 to 4 months. Zika was about a year, you know, and Ebola in 2014 was a little bit less than a year, but these outbreaks would happen really quickly. And our ability to react to them was always sort of stymied because we were always essentially playing catch up. And it takes months for these treatments to be available, as you've seen, right? The fastest vaccine ever developed prior to the SARS-CoV-2 vaccine that you all are receiving was measles, and that took 4 years. What we were able to do with SARS-CoV-2, not we personally, but just medicine in general, was on the order of a miracle. They got this thing out there in a year, and it works very effectively. But treatments, in order to really stem the tide of what we saw in New York City back in March, treatments have to be available within the order of a month, or else you're going to have a period of time where there's essentially no protection. And that was the really scary portion of the pandemic, where people were going into hospitals and there's literally nothing that we can do for them, right? Because the vaccine is still a year off and there's no treatment. So, how do we go about solving this problem? And this is where passive antibody therapy comes in. So the antibodies that Atomab makes could help stem the tide and offer a stopgap for these particular infections. And so these antibodies themselves are much easier to collect than developing a new vaccine. They're very safe because they're your body's own proteins. So you're not going to have some sort of issue where you are, in general, going to make a response against them and have an allergic reaction or something like that. But the problem is that the discovery and development of these antibodies still takes a multi-year process. And so her question was like, well, how is it that how would we ever be able to get to a point where you'd be able to have an antibody therapy that would still be within that window where it can have an impact and really punch a hole through what would be an emerging epidemic? And so if you were to overlay the timelines of just like discovery and development and testing and then manufacturing, this still doesn't work. This is still problematic. I mean, with SARS, that was about 3 months. You wouldn't even be out of animal testing before you would have the pandemic end, or the epidemic end. And you certainly weren't able to do anything to help anyone in the meantime. So we need to fix this. We need to find a way to anticipate what might be coming down the pike. And so the alternative approach that she offers, and others are offering now, is to try to develop antibodies that have the ability to recognize not only just the thing that has emerged, but previous variants of it, right? SARS-CoV-2, we have experienced in some way in the past, and that was SARS back in 2022. I'm sorry, in 2002. Whoa, sorry. These are things that have variants in nature. They have usually prior experience. And so what medical science is sort of consolidating around is the idea of creating broadly neutralizing antibodies to given viruses. So a broadly neutralizing antibody to influenza, for example, would be an antibody that recognizes this year's influenza, but also 1918 pandemic influenza and everything in between, right? Anytime the virus makes a move, the antibody still has a way of recognizing it. So then what you can do is start to break things down by disease class. And that makes it a lot easier to try to create stockpiles of drugs. So say when an Ebola outbreak happens in West Africa, it doesn't really matter what mutations might have occurred or where this virus had emerged from, because the antibody that you've made is still able to bind and protect. And this is a really, really powerful tool, and it's likely one of the ways that we're going to be able to prevent what we're seeing today. And so Laura and some of her colleagues at the United States Army Medical Research Institute for Infectious Disease, Scripps Research Institute, and Albert Einstein College of Medicine had written grants around actually trying to develop this pandemic preparedness. And we are working on it. What was unfortunate was we chose the wrong viruses. So, we're working on things like pontavirus and different hemorrhagic fevers that are emerging in the developing world. But at the time, we weren't working on coronavirus. And so Atomab and Laura, led by Laura, went about trying to plug in what we do with the Atomab yeast system with how the body is actually able to respond to viruses. And so this is taking in vivo, meaning in life, diversities, so antibodies deriving from you, and then using the Atomab system to try to do rapid characterization and rapid production of pharmacological leads that would actually be scalable in, for protection of individuals. And so she did this by acquiring blood from an individual who had been infected with the original SARS virus back in 2002. And so this person, we got a sample from the National Institute of Health, the NIH, headed up by Tony Fauci. And so they were able to get a blood sample to us of this individual who had been previously infected, and then using blood isolation technologies, we were able to take out the individual B cells, so one B cell at a time, and essentially stain them like you would painting them with the SARS-CoV-2 entry molecule. So the same thing that was used for the vaccine that you guys are experiencing now, we used for trying to fish out B cells in the body, in the blood, that actually bound to this viral target. And this is what it actually looks like. So you have your SARS-CoV-2 proteins, and then this population here are the cells that are able to bind specifically to the target of interest. These cells are then individually put into plates for amplification, so DNA amplification. And so you have one well that will actually contain only one cell. And this is something that's still amazing to me that we can even do it, but we have ways of sorting these B cells into 96 well plates, so these blocks, and you can individually sort them one at a time and then amplify their existing the antibody genes. And then it can plug really nicely into our discovery platform here at Atomab, where these antibody genes can then be essentially shoved into our yeast for then production and later characterization, and then finally optimization, like I've talked about. And so she and the team here did that. And so we took blood from a convalescent donor, so somebody who had previously recovered from the original SARS outbreak. And then we sorted for B cells that are able to bind to the new virus. And this is trying to pressure the discovery of the drug to something that would be broadly reactive to all coronaviruses. And so this was this was done in like March. So during the time where everything was shutting down, we were getting samples in and doing the sorting. Well, I wasn't, but other people in the lab would, to try to make these antibodies against the SARS-CoV-2 protein. And then once they had discovered them, they further optimized these antibodies to bind very, very, very tightly to a variety of the SARS-CoV-2, SARS variants, I should say. And so this has now become a drug which is in clinical trials, which we're calling Adagio 2. And so Atomabs spun out a company named Adagio for the production of these antibodies. And so this is actually what it looks like. So these are our antibodies. So what you're seeing up top are pictures, actually, of our antibodies with the viral protein. So in gray is the viral spike, the thing that you've been immunized with. And the coloring isn't real coloring, we highlighted them here, but the color portions are actually the antibodies that we've made binding to that thing. So this is a picture of this happening, zoomed very, very, very far in. And structurally, if you were to then do molecular modeling, you can show what this would look like. And our antibodies are binding to the entry site for the virus. So you might have heard about ACE2. This is this receptor that's in your lungs that the virus is using to uptake the virus. Or I'm sorry, yeah, the virus is using to get into the cell, I should say. Our antibodies are able to bind and prevent that interaction. And so they're binding specifically to this ACE2 binding site on the viral protein. And so this is just the zoom in of that with Adagio 2. But this is important because that means that these antibodies are able to actually neutralize the infectivity of the virus. And we found an antibody that sits outside of the binding site of all these other known clinical antibodies. So this is, for example, the Regeneron antibody. And then you also have the Eli Lilly antibody. They're all binding in a very similar manner to each other. And our antibody is actually sitting away from that. And that's important for this reason. So our goal was trying to find antibodies that bind and neutralize not only SARS-CoV-2, but other versions of SARS. So if SARS were to ever emerge again, our hope would be that these antibodies would be able to capture that virus and neutralize it and basically prevent you from having any symptoms of disease. So this is just looking at the ability of these antibodies to neutralize the virus. And so in blue here, dark blue is Adagio 2, that's the antibody lead. And these other colored dots are just other clinical antibodies. So antibodies from Regeneron, antibodies from Eli Lilly, antibodies from Merck, whatever. They're all here. We tested all of them. And so you can see that all these antibodies bind fairly well against SARS-CoV-2, which makes sense because they're all designed against that particular virus. Where it gets interesting is when you start to compare against other variants of SARS coronavirus. And so in blue is Adagio again, and then we're testing against the original SARS. So this was the guy that was circulating back in Hong Kong and Toronto in 2002. And then we also have here bat coronaviruses. So there is an animal reservoir for this disease, and it's likely that because of the factors that I talked about with globalization, climate change, all those things, and we think that this actually came from one of those wet markets in Wuhan, China, that the ability to neutralize circulating variants of the virus that exist in animals is going to be critical for trying to make a broadly neutralizing antibody in the future. And what's really cool and heartening about what we've been able to develop here at Atomab is that this antibody is actually able to bind and neutralize with very high potency against not only SARS-CoV-2, but these other previous epidemics or potentially future epidemics. And you'll notice that these other antibodies don't do that. So in this graph, down is good. So the lower the number, the more potent something is, meaning the less of it you have to give in order to have it have an effect. And what you see is that these antibodies do a really, really good job of doing that. And this happened like so fast. So it's been a whirlwind of the year, I'm sure for you and for us. But with the power of the system that Atomab has made, combined with our knowledge of B-cell immunology, we were able to make a drug in less than a year. So, and this drug could then be used for future pandemics. So what we want to do is make things that anticipate what's going to come, just like the immune system does. You're creating anticipatory receptors. We're going to create anticipatory drugs. So on March 19th, we got our sample in from the NIH. So the Vaccine Research Center sent us the SARS sample. On April 9th, they started to do the sorting of B cells. And on April 14th, we actually had antibody in hand that we were able to then test to make sure that it was actually neutralizing the virus. In May, so just like 2 months later, we had performed optimization of antibodies. And this was done by one of the scientists in my group. And then those antibodies were then sent to our pharmaceutical manufacturer in China, where they were entering into cell line manufacturing development, so scaling it up to be a large enough size where you could begin to conduct clinical trials. And then in December, this got sent to the FDA and filed and approved. So this is actually, you can go on to clinicaltrials.gov and you can see the Adagio antibody there. And so this was posted it was finally posted in March, but the filing occurred in December. And then the first in human trial went into people in February. And now, and so the government's always a little bit slow, but yeah, so it's online in March, but it's in phase 1 clinical trials as we speak. And it will likely be in phase 2 clinical trials sometime next month. And so this is.
Hey, Noel.
Yeah.
I know you.
Was I muted the whole time? I was muted, wasn't I?
No. You've got a hard stop in a couple of minutes. So
Yep.
Can we stop here and
Sure.
get a couple questions in?
I was at my end.
Oh, good, good. Noel,
Sure.
folks, Noel informed me that he had to leave at 12:50. So that was great. That was great. Jinya, your hand's up. Throw a question at Noel.
I think
I
this,
saw you writing notes.
I was taking frantic notes. I think it is so exciting. And I was taking notes hoping that we have recorded this
We
session.
have. We have.
Okay, I two questions. The first is, where is Atomab? In
Atomab is in Centera.
Centera.
So, yep. We're behind the RVC. If you know where that is.
And my second comment is that I happen to have two daughters who've been struggling with cancer, and noticing what's happened when they've had to do chemotherapy and all of that, I just was mesmerized by the fact that this will really help in terms of treatment.
It is incredibly heartening to sit in our lab meetings and you see where medical science will be in 5 to 10 years. And you just see the number of things coming through, all the different cancers and neurodegenerative diseases, viral infections that we're working on at any given time is dizzying. And it's really, really exciting to be a part of.
And what I hope is that the information will really be transmitted widely, because I think one of the problems I've seen with medical stuff is that somebody's really good at what they've developed, but somehow the rest of the country doesn't know about it. So
yeah,
hopefully you're doing a great job of passing on this information.
Yeah, we try our best. I mean, I hope I did all right with you guys. But the dissemination thing is something that's of great importance, and I think the pandemic has actually, for all of it being terrible, it actually has aided our ability to talk medical science with the general public, because everyone now knows about vaccines. Everyone is hearing about viral variants. Everyone's hearing about monoclonal therapies and drug treatments, and people now have a lot of questions, and it's really good to, I get my mom and my grandmother, they usually send me like multiple people per week who I have to talk to about what's going on in the world.
Yeah.
And so it's, it's a really helpful thing, because the more people that know about the science, the more pro-science people become, right? And I think that it's something that's very critical nowadays, because the only way that we're going to stop this pandemic and also prevent future ones is by having an educated public who care and are passionate and want to invest in this work. This is done by a private organization, but we stand on the shoulders of giants, right? All of this work is possible because of government funding. Everything that, and I am here, I'm a product of government funding. My PhD was created because Eisenhower wanted to make sure that we could beat the Russians into space, right? He came up with this program, and that's how I was able to go and get my PhD. Like, all of these things matter. And so to make sure the next generation is able to create the great therapeutics and great drugs, great discoveries, go to Mars, whatever, it all happens through communication and making sure people are aware that these things matter.
Thank you. I'm sure you are excited getting up every morning because you feel you're doing something really, really worthwhile. So
yeah, this morning I wasn't, because I stayed up late to get my vaccine time, and now I'm really tired, but most days I am.
Thank you.
Thank you. Noel, I see that it is 12:50. That was what you told me was your hard stop, and I want to respect that. So
yeah, if anyone has I can
questions.
take 5 more minutes if anyone has any other questions.
That's great. That's great. Heidi, you've had your hand up for a while.
Thank you. I have 2 quick questions. I had more, but I'm going to limit it to these. Number 1, can Atomabs products be manufactured in the United States? Because we're finding it more and more important to
yes,
be self-sufficient here due to a lot of political.
And that's actually, yes, we are having actually conversations with the US government about doing that. I will say, so that was specific to the Adagio 2 antibody, where it was manufactured in China, but most of our drugs through pharma partners, they're like in New Jersey, right? So there's a lot of drug manufacturing down there. This was sort of just a quirk of the relationship of how everything came to be. Because you have to find a factory floor that has space for you, and that was the one that we could find.
And my other question was, you briefly mentioned allergic reaction, and it's earlier on in your presentation, I made a note about, you talked about the binding process. So there's a binding process and a falling away.
Mhm.
And what your goal is for it to fall away on a longer timeline. So you want to increase the timeline. What if somebody has an allergic reaction though? And how do you have antidotes?
We do, yeah. And actually, so that's what my group did for all of this process, but I didn't actually talk about it because it's not as exciting. So we made something called an anti-id. And so an anti-id is an antibody that binds to the other antibody.
Yeah.
And so we made something that was very high affinity. And so say somebody's having an anaphylactic response, you could give this to them and it would neutralize the effect of the antibody that you're delivering because it binds specifically to the binding site of the antibody. So it can no longer bind to whatever might be causing the allergy response.
So do you know if the vaccine sites are, have access to the antidote on site when they're giving? Because some people are having, I mean, it's a very, very small, tiny, tiny percentage, but some people are having really violent reactions quickly after they have their vaccine.
Yeah, so two things. So the antibodies that we're making, so we haven't seen instances of this for the Adagio causing any problems in people. And that's likely because it's not binding to anything on you, it's binding to the virus. The vaccine reaction that you observe is likely a background reaction to a component in the vaccine. And I should say to this group, that is something that has happened, but it happens incredibly infrequently. And in all cases so far, they've been able to avert any massive side effects by just administering, I believe they're giving epinephrine to people when that happens, but we're talking in the order of like 1 in a million.
Yeah.
Well, yep.
Thank you.
Yeah.
Wayne, did you have a question? You're on mute.
You're muted.
Wanted to know how you decide what your next target is going to be.
Yeah, well, and that's, you know, because we missed coronavirus, didn't we? That was discussions about, we have only limited bandwidth, right? And so this is why we need more scientists working on these things. We can only do so much in our shop, and we need other shops to be working on them, other university labs. And we picked, for the initial studies that we did when we were trying to come up with pandemic preparedness, we picked targets that we thought would be potentially useful for the United States. I mean, hantavirus is something that crops up throughout the US. I mean, you've seen it in Yellowstone, we have it here, and it's very, very dangerous. And so it was kind of a choice of where we think we might get the best bang for our buck. And unfortunately, we picked wrong for coronavirus, but you know, going forward, we would hope to be able to cover more of these now that we kind of have proven the point of the importance of this.
Yep.
So, but now I do have to go. So
Thank you, Noel.
yeah,
That was
thank you very
that was
much.
really fascinating. Thank you very much. Really appreciate it.